that, we can move to Kari Neier. Kari did her PhD in Michigan and is currently a postdoc at UC Davis, doing omics for neurodevelopment. So with that, Kari, take it away.
Thank you. Yeah, so thank you to Monica and to Sophia for organizing this, and to UA for reaching out to me to invite me to give this talk to everybody. So I'm going to be talking about the role of the epigenome in influencing the risk of neurodevelopmental disorders, similar to UA's talk, but I'm going to be talking a little bit from a different perspective because my toxicology background has really led me to be interested in how exposures to chemicals influence our epigenome, thereby influencing disease risk. And so I'm going to be talking mostly about these chemicals called polychlorinated biphenyls, and they're abbreviated PCBs. So I'll be referring to them as PCBs throughout the talk. They're persistent organic pollutants. And basically what that means is that once PCBs are released into the environment, it takes a really, really long time for them to break down. So they remain in the environment for a really long time. And there are several different actual congeners of PCBs. And basically they have this structure here with the two benzene rings connected together, and then any number of chlorinations on in different conformations on these different ring positions, and that makes up the different congeners of PCBs. They were used historically in a lot of electronic material, and as well as in solvents. However, the explicit manufacture of PCBs has has been banned since the 1970s, but they're finding that more recently PCBs are being released as a byproduct of producing paints and pigments in paints. And so PCBs are actually still being released into the environment, although not purposefully. They're more of a byproduct. And because they remain so long in the environment, we're actually still exposed to PCBs that have been released into the environment decades ago. And developmental exposures to PCBs, or exposures during the period of development, are associated with adverse neurodevelopmental outcomes. And so epidemiological studies have actually linked developmental PCB exposures with impaired executive function, psychomotor function, attention, learning, and memory. And they've also found a potential increased risk for developing attention deficit hyperactivity disorder, ADHD, and autism spectrum disorder, which UA gave an introduction about earlier. And so the epigenome is really at the interface between the environment and the genome. And so I'm not sure if anyone here has an environmental health or toxicology background, but as somebody with a toxicology background, I was really interested in learning about ways that the environment interacts with our genome, and the epigenome is essentially the quintessential method for that. And so the reason for how the environment really interacts with the epigenome is that if you think about it, the epigenomic marks such as DNA methylation or histone marks like histone acetylation and histone methylation, these are actually influenced by the environment. And so they will respond to different environmental stimuli, including oxidative stress, which UA talked a little bit about, and as well as environmental chemicals. And so because of this, it's really an interesting method to study how exactly our genome is responding to environmental cues and environmental stressors. The epigenome also plays a really critical role in neurodevelopment. And so I'm just showing kind of a schematic of how different cell types in the brain develop and are differentiated from neural stem cells. And essentially, as stem cells are differentiating into their final neurons or oligodendrocytes or astrocytes, their epigenome is really in flux here. And so as the brain is developing, the epigenome is really rapidly changing the conformation in order to change chromatin states to become kind of their final state in a differentiated cell. And therefore, during development, when all of these marks are being removed and set back on, this is a really critical period where if we're exposing animals or humans to chemicals during this developmental period, it really could have long-term effects on our epigenome. And so this is another reason why the epigenome is really interesting to study. And then also this makes the period of in utero development a very critical period to study for chemical exposures. One of the key questions that our lab has is how do we really study the brain epigenome in humans? So UA presented some really great data looking at the placenta epigenome in humans. And the reason, obviously, probably why we don't do a lot of work in brain is because it's very difficult to get brain tissue unless it's postmortem. So for some of the big cohort studies where we're measuring exposures in human beings, we're not going to be able to actually get DNA from the brain tissue. And so that's why the placenta is a really attractive tissue to use, and especially if you care about exposures that are happening during development, because the placenta really regulates the fetal maternal interface. And UA already brought this up, but two of her previous studies are really good examples of how the placenta methylome really carries signatures of neurodevelopmental disorders. So UA found previously that looking at whole genome bisulfite sequencing data and looking at the DNA methylome, you're able to really classify a typically developing by autism spectrum disorder child just by looking at the methylation levels in the placenta. So that's really intriguing and brings up the possibility that we could use the placenta as a potential marker of what is going on in the brain, especially within the context of neurodevelopmental disorders. And so we wanted to, kind of further understand the relationship between epigenomic signatures that we see in the brain and those that we see in the placenta, so that we can get an idea of how well the placenta is really representing what's going on in the brain. And so what we did is we designed a mouse study so that we could actually look at brain tissue as well as placenta tissue from the same animals to get a better understanding of what's going on. So in this study, what we did is we looked at developmental exposures to human relevant mixtures of PCBs to see whether or not these developmental exposures result in different behavioral phenotypes and as well as molecular signatures of neurodevelopmental disorders in brain and placenta tissue. And I wanted to mention that this mixture of PCBs was actually determined by measuring PCB levels of the mothers that were in the human cohort that UA has worked on. So this should be a pretty human relevant concentration of PCBs. And so previously in Dr. Pam Lyne's lab, who we are fortunate enough to collaborate with, they found that the exposure during development to PCBs results in behavioral phenotypes in early life of the animals that were exposed in utero that are ASD-like. So for example, at postnatal day 7, which is really early postnatal day life for the mouse, they saw a decrease in ultrasonic vocalizations at all of the doses of PCBs tested. And then later on, as the mice were growing up, they found an increase in repetitive behaviors at the low dose and a decrease in sociability at the low dose in the male mice, but not in the females. So some really intriguing findings here showing that developmental exposures to PCBs might be influencing autism-like phenotypes. So we wanted to ask the question, do PCBs actually alter DNA methylation in the fetal brain and in the placenta, and how do those relate to one another? So we used whole genome bisulfite sequencing, essentially the same sequencing technology that UA used for her human samples, and we measured whole genome methylation in just a group that were exposed to vehicle and a group that were exposed to 1 milligram per kilogram day of PCBs, which is a medium dose with respect to some of our other dosing studies. And then we measured this in both the brain at embryonic day 18 and the placenta collected at embryonic day 18. In addition, we also measured the transcriptome, and I'll be talking about that in a little bit later. And I actually, so somebody had a question about the WGBS sequencing technology for UA. And so I actually am showing just really quickly some of the bioinformatic methods that our lab uses in order to assess DNA methylation and look for differentially methylated regions. This pipeline that I'm showing you here is DM-RICHR, which was developed by Ben Laufer, who is currently a senior scientist at Genentech. And essentially what he does is filter CPG sites for coverage. They do a sex check to make sure that we've called like the sexes properly. Then he calls DMRs or differentially methylated regions, or large DMR blocks, which are on the level of tens of thousands of base pairs. And then DMRs are annotated to the genome. Also, the methylation values across these DMRs are smooth using BS smooth. And essentially you'll get a DMR report, gene ontology, enrichment testing, plots and heat maps, and you can also do some machine learning. So just a quick plug for DM-RICHR if you have whole genome bisulfite sequencing data. And also just to answer the previous person's question about our sequencing methods for bisulfite sequencing. The last time I checked our bisulfite conversion rates, I think that they were upwards of 98, 99%. So they're very high. And so after we called DMRs in the brain and the placenta, what we found is that they distinguish developmental PCB exposure in both tissues. So here I'm showing you the placenta tissue on the left, the brain tissue on the right. The female tissue is always on the leftmost of those panels, and then males, and then females, and then males. And so here on the left, what you see is placenta. Placenta tends to have a very different sort of methylation pattern overall than brain, where you have a lot of these, like the percentage of methylation tends to be around like 50%, as opposed to having like that bimodal distribution where you have a lot of very lowly methylated regions and a lot of very highly methylated regions. And what we found overall is that really the samples clustered by whether or not they were developmentally exposed to PCBs. So the blue is control and red is PCB exposed, and you can see by these heat maps that they are really quite distinguished here. And this is for both placenta and brain. So this is really interesting because it looks like both tissues are really affected by these PCB exposures. We also found that DMRs were enriched in bivalent chromatin. This is really interesting because bivalent chromatin is kind of, it's more akin to being a poised state where a gene is could potentially be turned on if transcription factors bind to it, and additional epigenetic modifications are made. So when we see that these exposures are changing bivalent chromatin, that kind of indicates to us that the changes that we're seeing are happening at regions of the genome that could have implications for gene expression. We also found that fetal brain DMRs were enriched for neurodevelopmental processes. So females and males had slightly different biological processes that were impacted by developmental PCB exposures, but they were relatively similar. Some of the top processes that we saw that were different were neuron differentiation, cell projection organization. We saw a lot of synapse and postsynaptic membrane for the localization, dendrite for localization, and then ion channel activity was coming up also, and ion binding quite often as well. And so A lot of these pathways that we found coming up are pretty typical for what has been seen in classic PCB toxicology studies. So this was reassuring that we were seeing these signatures in the fetal brains as well. But what was really exciting is that the placental DMRs were also enriched for neurodevelopmental processes. So even though this placenta tissue is not the brain, we still are seeing signatures of neurodevelopment coming up as being impacted by PCB exposures in the placenta. So some of the top terms that you can see here are nervous system development. At the very top, cell adhesion, which is also really related to neurodevelopment, synapse, cell projections, neuron projection. And so this is really interesting. And this is the first kind of clue that we had that, oh, the placenta methylation might be telling us something about how PCBs are impacting neurodevelopment. We also found that a subset of differentially methylated regions directly overlapped by their genomic coordinate across placenta and brain. So this EV observed here is the number of overlapping regions that we found between placenta and brain in total. And it was far above like what you would expect based on chance alone, based on our permutation testing. And so this wasn't just a random chance that the overlaps between the placenta and brain were occurring. Like this was something pretty significant. We also found that DMRs in the placenta and the brain annotated to many of the same genes. So you can see here that at least 521 genes were commonly annotated to DMRs in the placenta of both male and female and the male brain. And 424 were common to placenta male, placenta female, and the brain female. And so this is really telling you that there are a ton of genes that are being impacted by PCBs and are also involved in neurodevelopment that are being impacted in both of these tissues. And so when we then wanted to see the pathways that were enriched for the genes that were annotated to DMRs that were common across the brain and placenta tissue, and again, we see a lot of neurodevelopmental processes. And then we also see split robo signaling come up and Wnt signaling pathway come up. So these two pathways potentially could be molecular signatures that could be used as biomarkers of PCB exposure and neurodevelopmental outcomes. And then finally, we also looked for enrichment in different publicly available data sets for gene expression data for different types of mouse models. And some of the top hit mouse models are MECP2 mouse models, which are a mouse model of Rett syndrome, which is a really severe neurodevelopmental disorder. And so this was really interesting to us as well, that we were able to see this specific neurodevelopmental disease come up, and a signature of that was showing up in both placenta and brain. And so that data was all recently published in Cell Reports, but I am going to talk a little bit about some unpublished data. Hopefully it will be published soon, just because I think it's really interesting and exciting. So after talking a lot about the epigenetics and what we're seeing with respect to epigenetic signatures, I feel like the next question is very logically, okay, well, you saw differences in epigenomic signatures, but did you see any differences in gene transcription? And so we actually looked at RNA-seq data from 4 different dosing groups. So a vehicle control group, a low dose of 0.1 milligram per kilogram day of PCBs, a 1 milligram per kilogram day dose of PCBs, and a 6 milligram per kilogram day dose of PCBs. And this allowed us to kind of assess whether or not there was a dose response relationship. And it gives you a little bit more power to really understand how these exposures are impacting the transcriptome. So just really briefly, our differential gene expression analysis involved some pretty standard pre-processing with trimming, alignment, QC, and filtering. And then what I did is I stratified the data by sex and by tissue. And then I use limb of Boom with dream weights, which allows us to control for litter as a random variable. And essentially, what we were able to do is model dose as separate groups where you can contrast all of the groups against one another. So we can look at vehicle versus low dose, vehicle versus medium, vehicle versus high, low versus medium, low versus high, and medium versus high. But then I'm also able to model dose as a continuous variable. And so in doing this, I'm assuming that there's a linear non-monotonic or a linear monotonic relationship with, and monotonic just means that as dose goes up, you expect to see response to also increase. And that means gene expression changes could either increase or decrease with increasing dose, as long as it's kind of in a constant direction. So looking at these two different methods, I'm going to show you a couple of tables looking at the number of differentially expressed genes, or the number of DEGs in these different tissues. And always this number on the left here is going to be like the number of the number of DEGs in each group-wise comparison. And the number on the right here is the number of the dose response, the number of genes that had a dose response as modeled by the linear models using lemma. And so this is male brain. And what you can see is that the largest number of DEGs is in the vehicle versus the high dose group, which makes sense. And then there's also a similar number of genes that were coming up as having a significant dose response. And when you look at the DEGs in the high versus vehicle analysis and in the dose response analysis, there's a large degree of overlap. So over 3,000 genes were in common to both of those. And there were only 710 that were unique to high versus vehicle. And there was about 1,200 that were unique to dose response. And that just really tells you that the PCB related differential expression in the male brain was driven primarily by the high dose group. And so you can kind of see that by looking at these lowest lines, that the high dose group here in purple has usually the highest response, whether it be an increase in gene expression or a decrease in gene expression. And then looking at the female fetal brain, this actually shows a pretty different story. So in this case, the biggest difference is between the low dose and the high dose group, and there are not any differentially expressed genes identified at an FDR cutoff of 0.05 in the vehicle versus the high dose group. And there were only 5 that were showing up as having a linear dose response relationship. And so this is really interesting and different. And so I explored this a little more just by visualizing some of the genes that came up. And when you plot out these genes with dose going across the x-axis and expression on the y-axis, what you can see is that these are really not monotonic dose response relationships. So in most of the cases here, what you see is this very low dose group has a pretty different looking response than the medium and the high dose group. And so then I wanted to look at the placenta. And so what we found in the placenta for males, what we saw in the placenta for males is that there were lots and lots of genes coming up as being differentially expressed in the vehicle versus the medium dose group, the vehicle versus the high dose group, low versus medium, and low versus high. But there weren't very many coming up as being a traditional linear dose response relationship. And so looking more at that, what we found is that there was a high degree of overlap of the high versus vehicle and the medium versus vehicle. So basically that led me to think that, oh, maybe the medium dose and the high dose are both pretty similar to one another. And the low dose and the control are very similar to one another. And when you plot them out, indeed, that's what it looks like. So in the case of the male placenta, what you see is that the medium dose group and the high dose group have pretty big responses in changes to gene expression, but they're pretty similar to one another. So it's not really a linear dose response group, which is probably why the dose response analysis wasn't showing us anything. So really you see like these big impacts at the medium and the high dose group in the male placenta, and not as many response going on in the vehicle and the low dose group. And then when I looked at the placenta of female mice, again, we have a very different story. This doesn't look to be like a traditional dose response relationship either for the majority of the genes. The biggest differences are in vehicle versus low dose, vehicle versus the high dose, and the low dose versus the high dose. And so when I kind of visualize some of these in the female placenta, we see that some genes displayed this non-monotonic dose response effect that we saw similar to the female brain, but some of them actually looked like a little bit more of a traditional dose response relationship. So it looks like depending on the gene and the tissue and the sex, you see a different response to PCB exposures and gene transcription. And even though there were these really complex dose response relationships, there actually still were quite a few genes that showed dose response effects in both brain and placenta for males and females when you look at the overlapping genes here. So even though it's a pretty complicated story of what's going on with the brain and placenta, we're still seeing several of the same pathways are being hit. And so overall, what we found with this project so far is that it looks like the placenta methylome and the transcriptome are indicative of PCB exposure effects in the fetal brain. We think that this has a lot of implications for human epidemiological studies. I think that these findings also highlight even more some of the findings that UA has found in the placenta in her human studies, and really gives us confidence that what we're finding is relevant for neurodevelopment. And then another thing that we found is when you look at lots of different doses, you see many different responses. And males showed kind of a more traditional dose response curve in both the placenta and the fetal brain. Although the placenta was a little bit different, with the medium and high dose groups responding similarly and the control and low dose groups performing similarly. Whereas females showed more non-monotonic dose response curves in both the placenta and the fetal brain. But some of the genes in the placenta did show a little bit of a traditional dose response effect. So it's a pretty complicated story we're still kind of working out, but pretty exciting. And then I wanted to relate all this back to some of the phenotyping data that our group has found in this mouse model. So what I showed you previously is that the most pronounced changes in the males in the fetal transcriptome were in the high dose group, but the long-term behavioral effects were found only in the low dose group in males. So why would that be happening? So we don't know why, but I think that it's possible that when the animals are exposed to a very low dose early on in life, you see kind of an adaptive transcriptional response and mild to moderate changes in the epigenome. And then because of this, the epigenome doesn't undergo any sort of corrective repair to this toxic insult, and these changes to the epigenome remain. And then later on in life, you see an altered transcription going on at these lower doses. However, with the high doses, perhaps what's happening is that the body is recognizing this as an overt toxic event, maybe because of lots of aberrant transcription, lots of changes in methylation. And then the body says, hey, like we have a lot of misplaced methylation marks, a lot of misplaced chromatin remodeling. And so we're going to fix that, repair the epigenome. And so later on in life, after the exposure has stopped, there's a repaired epigenome. And so there's no or little change in transcription and methylation, and then no behavior phenotype. So we don't know if this is really something that's going on, but I think it's an interesting concept that would be exciting to explore further. And so with that, some of our future directions are, so we've only evaluated so far the DNA methylome in fetal brain and placenta at one of the PCB doses that we looked at the medium dose. But we want to look at all 3 doses. And so we're currently working on evaluating the DNA methylome at all 3 doses in these tissues. We also want to look at the methylome and the transcriptome at later time points. So I mentioned that some of the behavioral phenotypes that we see in mice are happening at postnatal day 26 or 32, but we measured the methylome and transcriptome at embryonic day 18. So it would be really great to examine the methylome and transcriptome at these later time points when the behavioral phenotypes are seen. We also are interested in looking at analyses at single cell resolution. So right now, when you get a DNA methylation readout, it's an average of all of the cells that are located in your tissue sample. And so it's hard to tell if your changes that you're seeing in DNA methylation are due to a change in the proportion of the cell type. Are they due to changes that are happening just in one single cell type? And so it'd be really cool to look at this in a single cell resolution. And then we also would like to implement some machine learning to predict PCB exposure based on methylation in the placenta, because that would be really exciting and really applicable and useful for human studies going forward. And so with that, thank you to everyone at UC Davis, especially Dr. Janine LaSalle, and everyone in the LaSalle lab, especially Ben Loffer, who did a ton of the bioinformatic analysis for the study. Thank you to UA for inviting me to give this talk today, and to the various other LaSalle lab members. And then I also mentioned that we collaborate with Dr. Pam Lyons' group in the vet school at UC Davis. She is an expert on PCB toxicity and has been an awesome resource, as well as Anthony Valenzuela, who is a scientist in her lab who helped with a lot of the mouse work. And then Rebecca Schmidt, who has done a lot of the human work along with UA, so that we can make sure that the work that we're doing is going to be informative for human health. And then my individual funding that allowed me to carry out a lot of this work through NICHD. And so thanks. With that, I will take any questions from anyone.